Okay, we are on our fourth installment of Chapter 3, all about genetics. Today we are going to be talking about inheritance and patterns of inheritance, and our favorite, one of our favorites, Gregor Mendel. So to start off, we're going to be talking about Mendel's laws. So as a reminder, Gregor Mendel was an Austrian monk. He experimented with pea plants, and he's sort of known as the father of genetics. He is the person that kind of gave us all of the laws that we use for typical classical genetics. So the three laws that he is responsible for creating is the law of segregation, meaning that alleles separate, so each gamete only carries one allele for each gene. The law of independent assortment, meaning that alleles get inherited, they get passed on individually from one another. Of course, this doesn't hold true for linked genes, but unless it explicitly tells you that it's linked, you can assume that they are unlinked. And then the principle of dominance, meaning that recessive alleles will be masked by dominant alleles. And then, of course, there are always exceptions, right? Codominance and incomplete dominance do not hold true with true recessive and dominant patterns of inheritance. So again, we're always going to be reiterating things that we talked about previously. So haploid gametes are the sex cells produced by meiosis, right? Males have sperm, females have ova or eggs. And gametes contain one copy of each chromosome, so they only carry one allele of each gene. When those gametes come together during fertilization, they create a zygote, and those zygotes will contain two alleles for each gene, right? They get one from mom and one from dad. Males do have an exception because they don't have a paired sex chromosome, right? You only get one X and one Y, but those are not the same size or shape, so they are not perfectly paired homologous chromosomes. You can categorize combination of alleles in three ways, right? Homozygous, meaning that the alleles are the same, heterozygous, meaning that the alleles are different, and hemizygous, which is what you would call a male for their sex chromosomes because they only have one allele for each gene on a sex chromosome. So inheritance, so here's some more definitions for you. So you have genotypes, which is the allele combination of a trait. Phenotype, which is the physical expression. So genotypes would be like the letters you would use, like big B, or little b. And phenotype would be the actual description, like black fur or brown fur. Complete dominance is the idea that one allele is expressed over the other, whereas codominance is where both alleles are expressed equally in a heterozygote. So you, in this chicken example, you have black and white as your two extremes, and then the heterozygous has both black and white feathers. And then incomplete dominance is not shown here, but incomplete dominance is when you have a mixture of the two in between. So if you had like a red flower and a white flower, the heterozygote of that would be a pink flower, right? Incomplete dominance. It's a brand new phenotype, hasn't been seen before. Now, for human blood, it's one gene, but they have multiple ways in which you see inheritance in here. So you have three alleles, so it's multiple alleles, A, B, and O. And then on top of that, you have this idea of dominance and recessive, right? A and B are both dominant to O, but A and B are codominant to each other. So you actually get a fourth blood type, which is AB, and that is when you have one allele for A and one allele for B. Now, the environment can also influence what phenotypes are shown. One example is hydrangeas. They can change their petal color depending on the pH of the soil. So the more acidic a soil, the more blue it is, and then the more basic the soil, it becomes more pink. Human skin color is also dependent on the environment. If you live closer to the equator, then typically you have more melanin pigment production because it helps protect you against sun exposure. Whereas if you live further away from the equator, like in the Netherlands, then you have less melanin pigment production because it's unnecessary, and so humans haven't evolved to have higher pigment production if you live in that area. Now, hopefully you guys remember how to do Punnett squares. I'm not going to spend a ton of time on this. This is when you are crossing a parental generation to get the first generation, which is called F1. That is your first bout of offspring, and then you can cross those offspring with each other and get an F2 generation, which is like the grandchildren in there. So the way you do this is you set up like that window pane square and you fill in the boxes by bringing down the alleles from the mom and the dad. Again, I'm not going to spend a ton of time on this, so if you do not remember how to do Punnett squares, it might be worth your time to either check out a YouTube video or meet with me separately. Now, predicted versus actual outcomes. So when you are using a Punnett square, you are calculating the probability. That's why that word is bolded, because when you do a Punnett square, that does not guarantee exactly what you're going to get. It gives you a probability that you may get like a 3-to-1 ratio or a 1-to-1 ratio, but it will not tell you that you will get exactly 50-50, right? You'll get close to that ratio, but it's just a best guess. It's a probability. Now let's talk about genetic diseases. We're going to spend a little bit of time on this in this lecture. So a genetic disease is caused by a mutation which will change a gene's normal cellular function. A lot of diseases have been identified, but a lot of them are rare. So they can be autosomal recessive, where you have 2 faulty alleles, and if you're heterozygous, that means that you carry the faulty allele, but you also have a quote-unquote normal allele, which is what you show. So you phenotypically would be considered normal, but you would be able to pass on a faulty allele to your children. If it's an autosomal dominant disease, then you only need 1 faulty allele because it's dominant, so it's going to show even if you only have 1 capital letter. And then if the disease is caused by co-dominant alleles, then you only need 1 faulty allele, but if you're heterozygote in this capacity, then you have milder symptoms because you have 1 allele that is co-dominant and faulty with another allele that is normal, and so you get like partial symptoms but not full-blown symptoms of the disorder. So we're going to talk about 2 disorders really quickly. So cystic fibrosis is on the left. It is an autosomal recessive disorder. It's caused by a mutation on chromosome 7, and if you have this disorder, it produces a really thick and sticky mucus which clogs your airways and ducts of the digestive and respiratory system. And so you can kind of see in the pictures here, you have a clear lung pathway, right? Because you do not have this thick and sticky mucus, whereas if you have cystic fibrosis, you can see this mucus and it's causing a lot of scarring in this chest x-ray. If you are heterozygous for cystic fibrosis, you will not develop this disease. Now we're going to flip over to the right side and we're going to talk about Huntington's, which is an autosomal dominant. This is caused by a mutation on chromosome 4, and it has repeating trinucleotide sequence. And while that gene typically does have some repetition, more than 28 repeats causes it to be unstable and amplify even further. So when the repeats exceed 40, the protein will misfold and cause neurodegeneration, which means that your brain will start to degenerate over time. Okay, and you can see that you have your normal brain with your normal protein, and then as the protein unfolds and changes and misfolds, then you have some degeneration happening in your brain. So moving into sex-linked diseases, these are diseases that are found strictly on sex chromosomes. Because the X chromosome is so much larger than the Y chromosome, they're usually found on the X chromosome. And because of that, they're significantly more common in males because females have 2 X's. So you can always kind of mask a faulty allele with a normal allele, but males only have 1 X. So if they have a faulty allele, they have to show that faulty allele. So a couple examples of sex-linked diseases is hemophilia, which is where you can't clot blood. And this is a mutation to the coagulation factors on the X chromosome. And then red-green color blindness, where you cannot discriminate between those two colors. And this is a mutation to the retinal photoreceptors on the X chromosome. Now, lethal alleles is another category. And so when you have the genotype from a lethal allele, it can cause an organism to die. It's present when it's in a homozygous condition. So if you're heterozygous, it does not cause you to die, only when you're homozygous. Because of this, heterozygotes are able to carry this faulty allele, and you have the ability to pass on this lethal allele because heterozygotes have it but don't show it. So an example of this is achondroplasia, which causes dwarfism. So if you're homozygous dominant for this, it's lethal. And so because of that, you would get rid of that genotype, right? Because it's lethal, so it doesn't happen. So that's not a viable offspring because you would not have an offspring survive that genotype. So because of that, the ratios are then altered. So instead of having a 3-to-1 ratio, you would have a 2-to-1 ratio where 2 of your offspring would show dwarfism and 1 would not show dwarfism. Now, epistasis is another idea. This is really, really cool. I love the idea of epistasis. So this is where one gene controls the expression of a second gene. So one example of this is mice and their fur color. So black fur is dominant to brown fur, right? So the capital B is dominant to the lowercase b. But if they have a homozygous recessive hair pigment gene, so a totally different gene, gene C, it doesn't matter what B's they have because that C will override the B phenotype and all mice will appear albino. So as you can see at the bottom here, even though they have capital B's in these, it doesn't matter because they have 2 lowercase c's, so it overrides the B's and forces all of the mice to become albino. This also happens in humans where the gene for baldness supersedes hair color. So if you have the gene for baldness, you're going to go bald regardless, and it's going to supersede having red hair or brown hair or blonde hair, whatever color hair you have. Okay, pleiotropy is another version of inheritance. So this is when one gene affects multiple traits. So if there's a mutation in this gene, it's going to be really severe because it's going to affect more than one trait, right? So for an example here, the beta globin gene mutation, which is for sickle cell anemia, affects all of this. So it also can affect eyesight, your liver, and heart attacks. So this is, it's bad. Pleiotropy is one to watch, and you need to be careful of inheritance with this. So this is why some people do tests to see if they have a predisposition to passing this on to their offspring. Now, X inactivation. This is obviously another version of inheritance. So this is this happens in females where one of the 2 X chromosomes is inactivated. So this just makes sure that females don't produce twice as many X-linked proteins as males because females have 2 X's and males have 1 X chromosome. So what happens is that one X chromosome that's inactivated is silenced and packaged as a transcriptionally inactive, and it goes into this structure called a bar body, and it just stays inactive, and so you only have kind of like one functional X chromosome. So let's talk about a real life example of X inactivation. This is my cat. Her name's Aurora. I call her Sweet Potato though. And her fur color is a direct result of X inactivation. So she's a calico, she is female, and her fur color is an X-linked gene. So her fur color is located on the X chromosome. Now, one of her X chromosomes is coded for black, and one of her X chromosomes is coded for orange. And you can distinctly see what cells in her body have the black X chromosome inactivated and have the orange X chromosome inactivated. And you can see that in the patches of her fur. And so, anyways, I'm just going to leave this up for an extra second so you can just like stare at her cuteness for a sec. But that is a perfect example of X inactivation. So mosaicism is another example of inheritance. This is the presence of two populations of cells with distinct genotypes within a single organism. So X inactivation in mammals creates a mosaic of genotypes linked to the X chromosome only. So it's not mosaicism. This would be if it was in a chromosome that is not a sex chromosome. Okay? So this is any other chromosome. And gene mutations. So mutations can be spontaneous or induced by exposure to external elements, right? So you can get mutations from radiation, chemicals, biological agents. Any of those can cause mutations beyond just accidental mutations in your DNA. Anything that increases the rate of genetic mutation is called a mutagen. So think of I didn't do it, but think when the U.S. bombed Japan during World War II, right? So the bombing of Hiroshima or the Chernobyl incident, there's been huge consequences from both of those from the amount of radiation that was released into the surrounding area. So you have increased amounts of cancer, reduced T cells, which means you have altered immune system, and increased thyroid disease. So environmental things that are happening around you play just as big of a role to mutations in your body. And some of these things you can prevent, right? You can prevent yourself from smoking cigarettes, which would cause a chemical mutation in your body. But other things are a little bit harder to avoid. So it's just good to be aware of what's going on and ways to be thoughtful in protecting yourself. So the last thing we're going to talk about is pedigree charts. And pedigree charts are just ways to visualize how traits are inherited from generation to generation. So in this first one, autosomal dominant, you can see that the parents must be heterozygous because one of the children does not have the trait. However, because the parents are both affected, and they have a child that is not affected, you know it can't be recessive, because if they are both affected, then they would only be able to pass on a recessive trait. So these are just ways to look at the pedigrees and be able to determine whether things are autosomal dominant or autosomal recessive. For this, you can again see, because both parents have it but one child does not, it's dominant, whereas for the recessive, you see the opposite happening. Both parents do not have it, but one of the children does have it. So again, this can't be dominant because if it was, then both of the parents would have it if they passed it on. But again, the parents must be heterozygous to be able to not have it but give it to their children. X-linked dominant, you can see that the woman has it and passes it on to her son, right? She gives the X to the son, so the son has it, and then the son has one X and gives it to all of his daughters, right? So all of the daughters now show it. So that's dominant. And then for the X-linked recessive, you see that the father has it over here, gives it to his daughter, but then the daughter gives it to his son, to her sons. Again, they only have one X chromosome, so if they get that faulty X chromosome, they must show it. And since the mother had two faulty X chromosomes, right, because she's showing it and it's recessive, that means that all of the sons would be guaranteed to get a faulty X chromosome. Now, when we're dealing with sex-linked inheritance and we're looking at pedigree charts, you typically can't confirm that they're sex linked, right? Although it's like highly suggested, right? It's like clear that there's some weird sex linked thing going on here. I can't confirm it, but I could infer that that's what happened, and I can suggest that this is a sex linked trait. So again, that's all for today. I will see you next time.